Good afternoon, ladies and gentlemen. My name is Dr. Asavri Sawant, and it's a privilege to welcome you to the episode 6 of series Amplification, Reimagining Diagnostics with Experts, on the title Decoding Omicron, which is being organized by Voice of Healthcare in association with Cipla Diagnostics. So for today's talk, we bring leading minds and experts together on this exclusive session. Now, I would like to call and invite Dr. Vaishali Gupta, Head of Medical Affairs Team at Cipla Diagnostics, to start the session. Over to you, ma'am.
Yeah, thank you, Dr. Asavri. So, good afternoon, everyone. I am Dr. Vaishali Gupta, Director Medical at Cipla, and it really gives me great pleasure to welcome you to this webcast on decoding Omicron. We are currently amidst third wave of COVID-19, and newly emergent mutant variant Omicron has spread globally. The objective of today's webcast is to understand from experts in the field on all aspects about this Omicron variant, including its impact on the diagnostics. We are honored to have eminent faculty members for this interesting discussion today. We have with us Dr. Anupa Dixit, madam. She's the president of Lab Medical Services at Suburban Diagnostic India, based at Mumbai. And Dr. 𝐬𝐩𝐞𝐿, madam. She's a professor microbiology at Wellcome Trust Research Laboratory, Division of Gastrointestinal Services at Christian Medical College, Vellore. I thank them for accepting our invitation, and it's really an honor to have both of them here for this interesting discussion. I hand over session to Dr. Anupa Dixit, ma'am, to take it ahead from here. Thank you so much.
Thank you, VOH. Thank you, Cipla, and thank you so much, Dr. Vaishali, for the short and brief, crisp introduction. Thank you so much. So, Omicron, we've been talking about this like for months together. Now is the time to decode it and understand it thoroughly so that we know what we are looking at and we are better prepared for tomorrow and the times to come. So this all started on 26th of November 2021 when the WHO Technical Advisory Group on SARS-CoV-2 virus evolution declared pango lineage B11529 a variant of concern and designated it with the Greek letter Omicron. It was first detected, as you all know, it was found in South Africa. It has been detected now in at least 57 countries by 31st of January. So this is where we are now with Omicron. And yes, what we know about it is that it has high number of mutations going beyond 50 plus. And specifically, the importance is the 30 odd mutations are seen specifically in the spike protein region, which is why there is a humoral immune evasion leading to increased transmissibility and the pathogenesis henceforth. Now, Omicron has spread like wildfire. As we all know, we have all experienced it in the month of January, and now it's tapering down, but it's still there. And now it's responsible for, it's also known as one of the reasons for the third wave in India. By our own testing data at Suburban Diagnostics, what we have seen is that the COVID positivity rate jumped from around 6% in December to 41% in the month of January. So this is what Omicron has done to all of us. Let's understand and decode Omicron further so that we are better prepared for tomorrow. And we'll be asking and interacting questions. So feel free to talk about it, feel free to ask and discuss your concerns and queries. So we have Q&A and the chat box both available for asking questions. So my first question to Dr. Kang. It's an honor to have her again for a discussion on COVID, and it's a delight talking to her and understanding more about virus and virology from her. So ma'am, how exactly is Omicron different from pre-existing variants? There are two parts to it. What are the major reasons of genome where the differentiating mutations are present? What is different about it? And which specific mutation have contributed to the ability of Omicron to be more infective and is also able to bypass or overcome the immunity of vaccinated individuals? So it'll be great help if you can answer this.
Well, thank you for that question, Anupa. And I'm afraid I'm not going to tell you the names of specific mutations, because with Omicron, we finally gotten to a stage where you can't remember every important mutation's name. I think the important thing to recognize with Omicron is the number of mutations and where they are occurring. As you mentioned, 30 plus mutations in the spike region. And these are located in the receptor binding domain, which is critical because that affects both the ability to bind to the ACE2 receptor as well as influences neutralization of the virus. So both the immune response and the ability to infect can be affected by mutations in the receptor binding domain. We also have a cluster of mutations in the N-terminal domain. And when you have mutations in the N-terminal domain, those can also influence a different class of antibodies that bind to the virus. So the effect on, as we know with Omicron, there are many antibodies that were available as therapies, which we can't use anymore. And that is partially because of the mutations that are in the N-terminal domain, as well as the ones that are seen in the receptor binding domain. We also have another cluster of mutations, the third major cluster in the spike region, and that is at the furin cleavage site. Now, this is important again for the ability of the virus to be able to get in easily. And if you have mutations there, it can affect the transmissibility of the virus. So this is what we are interested in. Its ability to spread, its ability to evade the immune response. Fortunately for us, mutations in the spike region do not necessarily affect the ability of the virus to cause severe disease. However, it does appear that because this virus has mutations that make it not dependent on TMPRSS, the protease that cleaves the spike protein and allows for the attachment and the integration of the membrane, what you wind up with is a virus that uses an endosomal entry pathway instead of using the binding to the ACE2 receptor and direct entry into the cytoplasm. So all of these are mutations that have affected the behavior of the virus.
So ma'am, does it also mean that Omicron shows any difference in the incubation period, as we have seen with the earlier variants and the earlier virus? And also the timeline of infection. Is there a change with Omicron?
So it's actually interesting that you asked that question, because this virus spread so quickly that in the early days, and now I'm talking about early days as in 4 weeks ago, we thought that there was a reduction in the incubation period with Omicron, and that the increase in spread was because of the shorter transmission cycle. So if you become infectious 2 days after you've acquired an infection, the chances that you will transmit to others are much higher. And we thought that that was going to be very different between Omicron and, say, Delta, where you had a 4 to 6 day transmission cycle. However, we now have data from the human infection model that was reported just earlier this week, which states that you can be infectious 2 days after you are delivered a challenge dose of the virus. And this was a challenge dose that was the D614G, or the grandmother virus. And if you get transmission in 2 days after that, you begin to wonder whether the Omicron transmission is really less than Delta, or have we not studied these as thoroughly as we should. So, but I'd like to ask you a question, Anupa, about what you are seeing as differences between the waves that we've seen? Your laboratory, I know, gets lots and lots of samples and has from the first wave onwards. What's the difference in patterns that you have seen? Numbers, the kinds of samples coming in, what you're getting in terms of CT values. I think you have a lot to teach us with all of that.
Thank you, ma'am, for this question. Yeah, this is quite close to me. So we've been doing COVID testing since 2020. That was in the month of March is when we started doing RT-PCR testing. Yes, we have lots of data with us. We have seen 3 waves. First waves, 20 and 21, we had second wave, and now we had this recent third wave. So we did a lot of studies. We also did an antibody study just to see how the trend of antibody prevalence is, how long it lasts. And also recently we did a comparison of all the 3 waves, just to get an idea, is there a behavioral changes seen with the time which has passed by? So what we concluded was, yes, we tried to compare the CT values of 3 waves. So our findings suggest that CT value of first wave was on the higher side as compared to the second and third wave. There was not much significant drop in the CT value as expected in the third wave. We did not find that. So that was one. So there was no substantial significance that we can attach to the CT value as such. Second factor, we try to analyze the predilection to the adolescents or the children population in the third wave. Is Omicron affecting the youngster more? So there was a very slight increase in number when we look at the children population getting affected in the third wave. So again, not a very significant number, so that we can talk about, yes, there is a predilection for the children age group. But third thing is very clearly coming out is about the reinfection part. So out of total 700 odd reinfection cases, which we picked up in our own data for the same patients whom we have tested over and over again, we realized only 1% of the reinfection happened in the first wave out of the total number that I'm talking about. And then we had 11% reinfection happening in the second wave, but substantially 88% odd reinfection happened in the third wave itself. So there were a lot of patients who were coming back to us with positive RT-PCR test. So these were the 3 very alarming and striking features which we wanted to study on, but only the reinfection part is one which we can establish when it comes to a third wave being different from earlier 2 waves. So that was our study. And yes, the positivity percentage, definitely we did a comparison of how far did we go with the positivity percentage on all the 3 waves. We touched around 30% in the first wave, the highest. Second wave, we had a positivity percentage, which was hovering around 22 to 24%. But the third wave, we went up to 41% of positivity of the total RT-PCR testing that we did. So yes, COVID has taught us a lot. It has also taught us to think and evaluate the data and see how trends are. And probably we can also contribute a bit to the social cause and make others aware of it. But yes, it's a study which is quite thrilling. In fact, we also did a study on our antibody response, where it was very interesting. Like post-natural infection, the antibody was consistently present throughout a year. And there was an increase in the value until 90 days. So it plateaued beyond 90 days of infection. And it got sustained for 9 months, which the value titer was pretty decent. So these kind of analysis, and the post-vaccine also, we did some study where what we found, the response, this maximum immune response was attained post-second dose of vaccine within a month's time, as compared to post-infection antibody, where the maximum titer was attained at 90 days of post-infection. Also, the decline of post-vaccine antibody was around 60% in 6 months, which was only about 25% when we saw our post-infection antibody titer. So these kind of studies actually, we have few studies going on also currently. So that's from my side on the comparison of 3 waves. But ma'am, what I also wanted to check with you is about, how do you think, we are just talking about humoral immunity, but what about the cellular immunity? Is that something we need to keep in mind when we look at the post-vaccine? In fact, post-vaccination and post-infection also. How do you see cellular immunity, which is not measured by any of the laboratory, playing a very important role in preventing the transmission of infection? I think that insight information is what we are looking at from you.
Well, if we look at the data that has come from the phase 3 efficacy trials, when you look at vaccines, what you want to try and understand is the vaccine protecting? And do I need to actually do an entire phase 3 clinical trial to tell whether a vaccine is protecting or not? Or is there a marker of protection that I can detect, or a correlate of protection that I can detect just by sampling people who have received vaccine? And if we look at the data from the phase 3 efficacy trials, it was quite clear that neutralizing antibodies were a population level correlate of protection. So the vaccines that gave the highest protection also had the highest levels of neutralizing antibodies. And if we go a step further, in the assays that are done in most commercial labs, what we measure is really binding antibodies. And it was found that the binding antibodies had a reasonable correlation with neutralizing antibodies. So you have protection, and that is demonstrated or associated with neutralizing antibodies. Neutralizing antibodies are associated with binding antibodies. And to come to your question of T cell assays, I'll take the example of Omicron. When Omicron first appeared, the first thing that you do is, because you've got samples from people who have been immunized previously, you look to see, do they have the ability to neutralize? Do they have the ability to bind? Because antibodies are readily available from those individuals. Very few places store peripheral blood mononuclear cells in the hope that they will be able to evaluate them against new variants. But when that was done for Omicron, we wound up with very worrying data. Even with the vaccines that were supposedly the best, the mRNA vaccines, there was a between 15 and 45 times drop in neutralizing antibody responses. And that was the huge concern. There was clearly immune escape at the level of antibodies. And would that play out as immune escape in terms of T cells not recognizing Omicron either? Well, the good news is that we found, and in very small studies, looking at people who had been vaccinated or infected before, that cellular immune responses are preserved, that T cells that are made by people who have received the mRNA vaccines recognize about 90% of the epitopes of Omicron. So when we see a lack of severe disease, it's very possible that it is the T cells that are preventing the replication of Omicron. The other thing that was important to look at was, when you look at T cell responses, you look at it two ways. You look at the distribution of cells and you look at the functionality of cells. And in terms of the distribution and the functionality, it appears that function is preserved. And that, of course, is good news for us. So an explanation of what we are seeing is that we are seeing reinfections and high levels of reinfections because this virus is replicating, is getting inside and is able to bypass the antibody-based immune response and infect cells. But then it's not able to replicate in those infected cells because of the T cell responses. So we get infected very easily, but we don't see as much severe disease as we saw with the earlier variants.
have a question from Nepal. So the question says that people are vaccinated with Virocell, that is an inactive vaccine, and are being given Covishield as booster. Does this cause any benefit? Also, what is the difference between Omicron vaccine, as they are stating, and will take it the path of getting vaccinated every year?
So this is a really interesting question, and the question of heterologous boosting is one that is being discussed very, very widely. Actually, if you've taken 2 doses of the inactivated vaccine and then you get a dose of Covishield on top of that, we've seen this data from Thailand, where they did studies where they had given the Sinovac vaccine first, and then they followed it up with the AstraZeneca vaccine. And that resulted in an immune response that was higher than was with the inactivated vaccine by itself. So much closer to if you had been vaccinated with the AstraZeneca vaccine. So based on those data, this is a strategy that has been used in many countries to use a viral vectored vaccine on top of an inactivated vaccine as a booster. We don't have real world effectiveness data, but the antibody data certainly supports such a strategy.
And how do you, how has the massive global vaccination drive affected the evolutionary pathway of the virus? Has it affected the genome diversity of the virus, or is it still the same?
So a lot of people have, especially people who are worried about the long-term effects of vaccines, raise this question. What does it mean if the world becomes immune to some extent and the virus is changing? Because when you vaccinate people, you're delivering immunity against an older version of the virus, and the virus that is circulating now is the newer version of the virus. Did we get the newer version of the virus because so many people were vaccinated? Well, first let's look at the first newer version of the virus, which was D614G. Let's look at the second newer version of the virus, which has circulated very widely, and that was the Alpha variant. Let's look at the third one, which was the Delta variant. D614G arose before there were vaccines. Alpha arose before there were vaccines. In Delta, India had just started the vaccination program. It wasn't covering much of the general population at the time that Delta emerged. So all of these viruses emerged before there was widespread vaccine use. Omicron is the one that has emerged after we've had widespread vaccine use. Now, when we look at what situation this creates, right? New viruses, antibody in people, that's actually like every other infection that we have with RNA viruses. Influenza does the same thing. Rotavirus does the same thing. People have antibodies, the virus continues to circulate. The important thing we have to remember is that for all of these, essentially, these are mucosal infections. And for a mucosal infection, being able to create sterilizing immunity is nearly impossible. You have to have local antibodies to shut down completely the replication of the virus. And right now, we are going for a systemic immune approach using injectable vaccines. So I don't think this is something we need to worry about immediately. There's certainly no evidence that vaccination has led to the emergence of new variants.
There's also one question which is pertaining to the vaccine schedule. So if somebody is infected with COVID and is mildly symptomatic, when should the booster vaccine be administered? Or for that matter, when should the vaccine be administered in that patient?
So I think, if you want the best possible immune response, separate your exposures to the virus or parts of the virus as much as possible. So if you can separate it by 6 months, very good. If you don't want 6 months, at least 3 months is a pretty good idea for separating infection and vaccination, because you want to give your immune response time to mature. And maturation, as you pointed out yourself with antibodies, when you have infection, you said the antibodies climb till 90 days. So that's a good benchmark to have, to say minimum 3 months.
And also we should keep in mind the vaccines. Why are the vaccines given? Is it to stop the infection or the spread, or is it to reduce the severity? I think this is the second question which has been asked. It's always about reducing the severity.
Absolutely correct. But okay, so let me turn this around on you, Anupa. What would it take to stop transmission? So would you like to discuss what approach we could take to stop transmission of a respiratory pathogen?
So in case of COVID, we definitely need to follow 3 W's and 2 V's. 3 W is wear the mask, wash your hand, and then comes your keep the social distancing. Maintain that distancing. From 2 V's, I take, you need to get vaccinated for sure. Vaccine is something which goes without saying, and that is going to mount our immune response and help us prevent spreading the infection and getting a serious effect of the disease. So that is the take home from my side when it comes to spreading the transmission. But what is your say regarding the detection of the test? How do we detect Omicron? What all do's and don'ts do you recommend when it comes to SGTF, which is a very known word which was roaming around, going on around in the month of January? Now we are hearing about this Omicron BA2 version, which has come, the sublineage of BA1, BA2, now BA3. There are 3 sublineages. What's your view on that? Is any data which can corroborate well with the BA2 now, which is more prevalent across as compared to BA1?
So there are some groups that have developed new PCRs that detect separately BA1, BA2. BA3 does not seem to be as important or spreading as much as BA1 did or BA2 is doing at the moment. You have to have either the specific RT-PCR or you have to have sequencing. When you look at the S-gene target failures, that was fine when the bulk of the strains that you were getting were BA1. But it's important to remember that some deltas also have S-gene target failure. So it isn't, even at that stage, we needed confirmation by sequencing. And I think being able to rapidly develop and deploy PCR-based tests would allow us to have much faster turnaround time if we wanted, for any reason, to be able to identify the variant that is causing the infection. And of course, in the case of the early days of Omicron, when it was co-circulating with Delta, it was important to know that in order to decide whether to use monoclonal antibodies or not.
So the most accepted and the commonly used method of diagnostic test is RT-PCR. And what we have seen is in RT-PCR, we target multiple genes and we detect the virus, such as spike, the envelope target gene, or maybe the nucleocapsid or the RDRP. It depends on the RT-PCR kit that we are using. We have various combination of genes which we pick up. So, however, we know that in Omicron, the S gene is heavily mutated. So some of the primers may lead to result indicating absence of S gene. So that is what we call as SGTF. For those who are not aware of this, SGTF is the S gene target failure. So as the particular S gene dropout along with the detection of other genes could be used as a diagnostic or a surrogate marker for the presence of Omicron, as you were mentioning. So definitely RT-PCR may not be the final diagnostic tool for Omicron detection. One has to undergo genome sequencing for the same RNA sample so that we can establish and confirm it to be Omicron. But since we are using kits which include other gene apart from spike for the detection of likelihood of missing Omicron variant is negligible. So very unlikely that we will miss out on Omicron positivity. Otherwise, SGTF may not work in cases of BA2 lineage because in BA2, we may not see S gene dropout, which means S gene will be picked up. And then, as you mentioned, we may not be able to differentiate BA2 with Delta. So we got some information regarding the sequencing analysis or the whole genome sequencing, which talks now BA2 lineage has come up as a very powerful viral sub-lineage, which is prevalent now in the society as compared to the BA1, which was earlier present. In fact, Delta has come down less than 10%. BA2 is now 30 to 40%, and BA1 is actually variants to come and be prevalent around. Is that what we are looking at, or is it BA2 is the last one? So how is it going towards, what is the way ahead? Need your opinion on this.
Well, obviously, we are going to continue to see variants emerging. We know this from other RNA viruses, that they are constantly evolving. And actually, if you look among RNA viruses, coronaviruses are among the slowest evolving viruses because they have a proofreading mechanism which smaller RNA viruses don't necessarily have. So we should be looking at lower circulation of coronaviruses, but we will also continue to see and therefore need to be able to track with rapid antigen tests, with PCRs, with genome sequencing, the new variants that arise. So I actually wanted to turn to another modality of testing and ask you, Anupa, about serological testing. How has your thinking on serological testing evolved with the waves and the variants?
So there are three types of testing, which the category can be. One is RT-PCR. We know we target the nucleic acid and we target the target genes and we pick those to actually diagnose. Second would be the antigen test, which actually targets the peripheral of the virus and which is also known as the outer protein. Majorly, it is nucleocapsid. We are not reaching the target genes, but we are reaching the nucleocapsid in particular. And this is where antigen tests come into the picture. Also, the serology test is the antibody test, which we do. Antibody test is not a direct measure of picking up a virus, but it also talks about whether somebody was infected with virus previously. But it is not the diagnostic tool for RT-PCR positivity or a viral positivity. So when we talk about the antigen test is majorly around nucleocapsid. And what we know of, the majority of the mutations that are happening or we have seen are targeting the spike region. So one thing is for sure, whenever we talk about a variant and we do an antigen test is not much affected by the presence of a variant. So it's still picking up all the variants, although the sensitivity of antigen test is little lower than RT-PCR test, which can go around 50 to 80%, but it's not 100% as what we talk about in RT-PCR testing as compared to RT-PCR. But specificity is very high. And yes, it's a very good indicator for getting the test done when somebody is symptomatic. But if symptomatic comes negative on the antigen test, we definitely recommend the RT-PCR in that case. And also when we talk about the serology test, which we talk about the antibody response, I think it's not affected at all by the variant because we are covering the spike, we are covering the nucleocapsid. There are so many kits available nowadays. You just need to know what you want to know. And the test is available. So specifically for nucleocapsid antibody, we have a antibody level detection. We also have against spike protein, which is specifically meant for the post-vaccine antibody level. So there are various platforms and various ways of detecting the antibody. That's not much affected by the variant that is going on around. So ma'am, one more question which majority are asking is again about the vaccination. So all the questions are related to the severe adverse effects of vaccination. What is your opinion on the adverse effects? Are there a significant number of deaths reported? So any information when it comes to the vaccination and any benchmarking on what type, although you mentioned about mRNAs are much better and far better when it comes to the efficacy in preventing an infection, but what are your views on vaccination as such? Also, do we need a specific vaccine to be coming up as we have new variants around? So is there a need for that?
Sure. So let's break that into two parts. And the first one on adverse effects of vaccination. I think one of the things that's really important to recognize is that the clinical trials that were done for vaccines were really pretty large, 25,000, 30,000, 45,000 people recruited into trials. And if you remember with the AstraZeneca vaccine, when the trials were going on, we were very worried about transverse myelitis and neurological symptoms with the AZ vaccine because there were 2 cases. Now, when the vaccine started to be used, because it was being used in high-income countries that have a good pharmacovigilance system, they were able to rapidly identify thrombosis with thrombocytopenia syndrome as a problem that was occurring at higher than expected frequency. Now, this is a condition that is actually known. And the fact that the pathogenesis is because of antibodies to platelet factor 4 is also known. Nobody completely understands why it occurs in people who have received the vaccine. But it may have something to do with the vector because you see a similar finding with the Johnson & Johnson vaccine also. It's estimated that globally about 1 in 65,000 people who receive the AstraZeneca vaccine might develop thrombosis with thrombocytopenia syndrome. Now, if that is the case, then there should have been hundreds of such cases in India. But our AEFI system was only able to pick up very, very few cases. And that tells us that our AEFI system has a long way to go. I think it's really important that as medical professionals, we emphasize the importance of safety surveillance, because if you have safety surveillance, then you know what to do. You can recognize the problem and you can treat it. I'll give you the example of Australia. Australia barely uses AstraZeneca vaccine. But in their thrombosis with thrombocytopenia syndrome, because of early recognition, mortality is 3%. So if we were to say 1 in 65,000 people develop thrombosis with thrombocytopenia syndrome, but even among them, only 3% will die, then that just makes all of safety surveillance worthwhile. If we look at the mRNA vaccines, again, once the vaccines started to be used in millions and millions of people, we noticed first that anaphylaxis with these vaccines is higher than with some other vaccines. For example, with influenza vaccine, you get anaphylaxis in 1 in about 800,000 doses, whereas with the Moderna and Pfizer vaccines, it's 1 in 400,000 and 1 in 200,000 people develop that. We also have in younger people this issue of viral myocarditis, especially in young males with the mRNA vaccine. What are the chances that we would be able to identify this in India? We just don't have the safety surveillance systems. The second thing you asked about was new vaccines and the direction that we are going in and whether we need vaccines specifically for variants. We know that new vaccines are being developed for Pfizer, for Moderna, for the AstraZeneca, and for Novavax, which are based on the Omicron strain. Will we need them? That's the question I can't answer, because we've managed quite happily through this Omicron wave without having an Omicron specific vaccine. So, but insurance policies are always good. So if we have the vaccine, should we at a later stage recognize that we need it, then we'll have it available.
Right. There's a long question which might take a while, but probably exposure to several laboratories. I think we will take some time to read this. It's a very long question.
So if you're referring to Sudish's question on exposure, and so I think everybody can look in the Q&A box at the question, where Sudish has pointed out that he's had known exposure to many people and he hasn't developed symptoms. And whenever he has tested, he has also again not tested positive. He's also got 3 doses of vaccine, and his colleague who got the booster dose developed symptoms and tested positive. So his question is he naturally somehow resistant to the virus? And is it because he has a very high level of antibodies, probably neutralizing antibodies, or was he infected and was the amount of virus that he had at below the detection limit of RT-PCR? So Sudish, since you tested many times and your RT-PCRs were always negative, I think it is very likely that you were probably not infected. The only way to know for sure is to look at your antibodies and to do both binding antibodies as well as neutralizing antibodies. Because even though they are correlated, it is not a perfect correlation. So the wider the net you cast to rule out exposure, the better it will be. Count yourself lucky, or you should be an example to others, because I'm assuming you always wore a mask.
Right. There's one more question or thought regarding long COVID. We all know what long COVID is, but what we have seen is with Omicron, there is a different variety of long COVID which has come into the news. And what's your take on this?
So I think there is a difference between persistent symptoms and long COVID. If we look back at Omicron, Omicron has been in India effectively for about 6-7 weeks at most. It's too early to come up with clear phenotypes of long COVID. So let's just see whether this is a prolonged course of recovery, prolonged convalescent phase, or whether this is truly the long COVID where symptoms persist for several months, if not years.
We've been also hearing about 𝐬𝐞𝐞, and the rounds of discussion were going on. And so what are your thoughts? Because my understanding says that it's not prevalent in humans as such. It has come from a bat known as 𝐬𝐩𝐞𝐞𝐿, if I'm not wrong. So what's your thought? Which way is it going? Is this the new Omicron?
No, this is just a coronavirus that has been found in bats that has made it to the newspapers. But this finding of coronaviruses in bats is something that has been going on for a really long time. When you look, you will find it. And this was found more than 10 years ago and has been circulating and hasn't crossed over into humans. And from the sequence data, we know that this is a virus that is unlikely to bind to the receptors that other SARS coronaviruses certainly are using for entry into cells.
Any other question from the audience? I think we have a few minutes left with us. So is there an immune basis test available which can differentiate natural infection versus vaccinated? So this question probably I can answer. Yes, we have a test and a platform which differentiates the post-vaccine versus the post-natural infected antibody level. Now, how do we do that is, we all know that vaccines are targeting towards the spike protein, more so RBD domain, so that these are the specific antibodies which are produced against the spike and the RBD. Now, when we know that a virus has multiple target genes like spike, nucleocapsid, then envelope, and others, if you are able to pick up others, we know that the virus infection was present. So, for example, if a test, antibody test, which picks up nucleocapsid, but it doesn't pick up spike protein, we know that the person has immune response post-viral infection, not post-vaccine. Vaccine is targeted specifically to the spike protein. But this is it's not a rule of thumb, but again, there are few viruses which also target nucleocapsid. So first we need to know which kind of virus was, which kind of vaccine was taken, and then we plan the testing accordingly. Like Covaxin will have both spike response and the nucleocapsid, but Covishield will have specifically spike response. So we can plan the post-natural infection antibody test basis the test which has been undertaken or the vaccine which has been taken. So it's doable. It's possible to pick up the antibody response basis the vaccine and post-infection. So I think the role of cell-mediated immunity response is what we have discussed earlier also, which ma'am has elaborated.
Anupa, I had a question for you. There has been a huge emphasis on the development of diagnostic kits in India.
Right.
So can you give us a description or a summary of the kinds of kits that you are using for molecular diagnostics in India and where they are made, and your reasons for choosing? Because I think for this audience, it's really important.
Right. So when it comes to RT-PCR diagnosis, when we started 2 years back, we had a very limited choice of RT-PCR kits available. And few of the kits were targeting only spike. Now I remember how spike came into the picture. They were targeting spike gene. And they were very expensive kits. It took a lot of time, processing time, and the copies that were picked up, which is the analytic sensitivity of the kit, is again very important. But over the period, in 2 years, I have seen such a great transition when it comes to the number of kits which are available, the improvement and enhancement technically. So the criteria that we follow, or any good laboratory will follow when it comes to the kit selection, would be primarily the sensitivity of the test, which is very important. We don't want to miss out and give a false negative report for a known positive. So sensitivity, analytic sensitivity, which means the cutoff, the minimum value that the viral load which can be picked up. So that becomes first criteria. Second is about the ease of processing. It's very important. It has to be user-friendly. It has to be compatible with the VTMs that I use, compatible with the extraction that I use. Because remember, RT-PCR test is not a very simple test to do. It is very intricate and requires a lot of stringent conditions and skilled manpower, of course. Now, third would be about the turnaround time. You cannot have a test getting done on a kit which takes a lot of time or hours together. So when we started processing 2 years back, we had kits which had a processing time of around, the PCR testing itself was 3 to 4 hours alone, as compared to it has come down to 1 hour. So there is a lot of improvement when it comes to the throughput and the turnaround time, which is very crucial right now. Fourth would be the type of genes which are targeted. I think this is a new learning which we learned out of Omicron primarily. We were not bothered much about the spike protein present or absent, but with variants, now we are very conscious about it. Like we should have multiple targets which should be available in the kit, so that even if there is a mutation in one of the gene target, at least other targets are picked up. So you don't end up giving false negative. So one of the criteria is good to have multiple target genes present for the detection instead of having or going for only 1 or 2 targets. So these are the 4 primary ways we can choose the kits. Of course, commercial and other things are not very important, but this becomes very, very important for us to take a judicious call. So there are a lot of kits available nowadays and good options. I also want to talk about few kits which are now targeted towards the variant detection as well. So there are few, ICMR has released advisory on purposive testing strategy for COVID-19 in India. So where they mentioned if Omicron or variant detection RT-PCR assays are recommended to be used in areas with limited genomic surveillance. This is what they're recommending. So going ahead, probably very soon we'll be having all the approvals ready for the RT-PCR, which will pick up variant as well, which are now readily available. And a lot of new kits are in pipeline as well. So good news from RT-PCR. Probably we may not end up doing surveillance and genome surveillance going ahead. Maybe RT-PCR will give the answer on variant.
I think that was tremendously helpful, Anupa, because there are so many people that do smaller scale testing than your laboratory. So good to hear from your experience. This has been a really interesting discussion. So thank you for the framing. And do we hand back to Vaishali now?
So we can conclude about Omicron. It is highly transmissible. This is evidence-based. It evades immune response. Ma'am can second me on this when it comes to vaccine or post-natural infection. Third is good news. It is asymptomatic and mild to moderate. It's very less in number, which means that it's predominantly asymptomatic by nature. I think these are the few good things about Omicron.
Thank you so much. Thank you so much.
I will hand it back to you.
Yes, thank you so much, ma'am. I thank Dr. Gagandeep Kang, ma'am, and Dr. Anupa Dixit, ma'am, for such a wonderful and interesting discussion and bringing out all great insights and facts about the Omicron variant, vaccination, the booster vaccination strategy, all kinds of tests, diagnostic tests which are available and which will be available, will be made available. And it was really it was really interesting discussion, and I'm sure it was very informative and useful to our audience. I thank our audience for spending their valuable time with us, and thank you so much for sharing all your experiences, sharing all your evidences and your own study results as well. Thank you so much, ma'am, both of you. And we are signing off now. Thank you so much.
Thank you.
So thank you, team.
Thank you, ma'am.
Thank you. Thanks, Rohit. We are offline, right?
Yes, ma'am.
Yeah. Thank you so much. Bye.